Hello friends, in my previous lecture we started with the various factors that can affect the quality of medicinal plant during the cultivation and after, that is post cultivation. In that we discussed that there has been a significant increase in the demand of medicinal plants or their products, and this in turn has increased the requirement for the raw material, which is always in the form of crude plant material. Majority of this increased demand of raw material is fulfilled by supplying the material that has been collected from the wild sources or the plants growing in their natural habitat. Then we also discussed what are the problems that arise when we collect the plant from their natural sources, and these problems were namely adulteration, irregular supply, non-uniformity of the quality, biodiversity depletion and genetic depletion. We also discussed that why cultivation of medicinal plants is important and why a lot of stress is being now given on the cultivation of medicinal plants rather than from their or their wild collection, and how cultivation basically helps in overcoming the problems that arise from the wild collection of the plants. And the benefits of cultivation were uniformity in the quality, regular supply of the raw material, then conservation of medicinal plant, utilization of modern technique was another advantage of cultivation, then development of the area where the plants are being cultivated were few of the advantages that we discussed. Then we started with the factors that affect the quality of the medicinal plant during and post cultivation, and we categorized those factors into the factors that affect during cultivation and the factors that affect post cultivation. We also categorized those factors under environmental factors and non-environmental factors. And we started with the effect of type of soil on the quality of the medicinal plants, and in that we discussed the effect of particle size of the soil, which eventually affects the water holding capacity of the soil, then pH of the soil, which also affects the activity of various metabolic enzymes that are involved in the production of the secondary metabolites of the plants. So friends, in continuation with the previous lecture, so the next factor I am going to discuss is the effect of salinity in the soil. Now salts in the soil, they mainly come from the water or by the natural withering of the rocks that are present in the soil. And the salts of sodium, calcium, magnesium, potassium and to some extent chlorine have shown to be the major role players in salination of the soil. Now every plant has its own requirement of salts for its growth or the production of secondary metabolites. And yesterday I also told you that there are certain plants which are vulnerable to a specific type of salt or a mineral, because most of these salts are nothing but the inorganic minerals or inorganic material that is present in the soil. And we discussed the example of digitalis, which is a calciphobous plant, and it does not grow well in the soils that are rich in calcium. But it has been observed that if digitalis is grown under moderate saline conditions or moderate saline soil conditions, then there has been an increase in the production of cardenolides or cardiactive glycosides, which are the main bioactive compounds of digitalis. And if we grow digitalis in extreme saline conditions or soils that are rich in salts, then obviously it will have an adverse effect on the growth of digitalis and production of the cardenolides. On the contrary, it has been observed that different species of mint, which are used for their oil or the essential oil, the oil production decreases when they are grown in a saline soil. In case of Marticaria chamomilla or the chamomilla plant, the production of phenolic constituents have been found to increase when that plant is grown under the salty soils. So this is basically how the salinity of the soil affects the growth of the plant, vis-a-vis the production of the secondary metabolites, and eventually it will affect the quality of the medicinal plant that is being grown. So one has to be very careful regarding the requirement of that particular plant, that plant will grow in which type of saline condition in that particular soil. So one has to be very careful while selecting or manipulating the soil conditions for a particular plant. Next we have is the organic content that is present in the soil. Now organic content is largely defined as the decomposed debris of plants or the animals, dead animals, or the animal parts or the dead animal tissues that are present in the soil. And we know that the organic content helps in the overall growth of a plant, overall development of the plant. And when we say development, it means the physiological development vis-a-vis metabolic development also. And depending upon the organic content present in the soil, specifically in the topsoil, it can be categorized into rich, intermediate and poor. And the topsoil, if they have organic content more than 5%, they have been categorized as rich soils, and the soils with organic content ranging from 0.5 to 1.5%, they are considered as intermediate soils, and below 0.5% soils are considered as poor soils. Now various studies have shown that the rich organic content present in the soil, specifically the organic content which contains amino acids or carbohydrates or lipids, that has shown to increase the production of alkaloids in specific plants. Because organic content, if we go for its categorization per se, then it can be categorized into living organic matter, which is mainly rich in carbohydrates, amino acids or lipids, and the non-living organic matter, which is mainly composed of humic substances like fulvic or humic acid. So if the soil is rich in organic content, different studies, as I told you, that it has shown that there is a specifically increase in the alkaloidal content of the plants that produce alkaloids. So organic content is another important factor that will affect the quality vis-a-vis the production of the secondary metabolite. One can easily manipulate the organic content of the soil by adding various kinds of fertilizers, manures, or even biofertilizers. When you are using chemical fertilizer, again you have to be very cautious that what kind of fertilizer you are using, whether it meets the requirement of the plant in question, which is under cultivation obviously, then only you can select that particular fertilizers. But because of the deleterious effects of the chemical fertilizers that are being used non-judicially by the farmers or by the cultivators, now a lot of stress is again now given on the biofertilizers, or which are mainly composed of the manures that are obtained from the natural sources, or development of microorganisms that will help in the overall growth of the plant. So one has to be very careful regarding the organic content of the soil. Another important aspect of soil that will impact the quality of the medicinal plant is its pollution. And in the recent years, we have observed that soil pollution is emerging as the major factor that determines the quality of the crop you are growing. And not only the quality, but also it also addresses, or it also relates to the safety aspect of the crop. And the soil pollution can be categorized largely into heavy metal toxicity of the soil and the abundance of pesticidal residues that are present in the soil. Now heavy metal toxicity, it occurs either with the air pollution or the water pollution. Now in air, we know that lot of industrial exhaust, lot of vehicular exhaust, it contains numerous heavy metals in that. And by virtue of these particulate matter or the toxic material that is being exhausted by the industries or vehicles, they remain suspended in our atmosphere, and they come down into the soil with the help of rainwater or acid rains, we can say. So all these toxic residues, they enter into the soil through air pollution with the help of water into the soil, and we know that majority of the nutrients or water is being absorbed by the plant through that soil. So they enter, these heavy metal residues, they enter into the plant system through the soil, thereby causing certain quality and more importantly safety issues with those medicinal plants under cultivation. Second was the pesticidal residue aspect of soil pollution. Now this we have seen in recent past that in order to increase the yield or in order to protect their crop from the attack of pest, there has been a non-judicial use of pesticides on the crops or the medicinal plants. And all these excess utilization of pesticides has led to the leaching of these pesticides into the soil with the help of groundwater. And again, by the nutrients that are being absorbed from the soil by the plant, these pesticide residues, they again enter into the plant system, thereby posing very strong, I will say, stringent, not stringent, but strong safety issues for those medicinal plants. One has to be very careful when they are cultivating a medicinal plant. So one should not cultivate these plants near any kind of industry where a lot of industrial waste is being thrown out of the industry, or that industry is exhausting so many gases, or near the highways or the roads which have high vehicular traffic. So one should be very careful while selecting the site for the cultivation of medicinal plants, because these factors also impact the quality. Now, apart from the different factors regarding the soil which we discussed, so once you select the suitable soil, you select the suitable conditions of the soil, the next factor is the quality of the seed which you will use to develop the crop. Now, if you are using a good quality seeds, then obviously you will get good quality crop which will be resistant toward different kind of diseases or infections. And the quality of the seed can be determined broadly by different attributes or different factors like physical factors or physical attribute, physiological attributes, genetic attributes, and storability. Now, the physical factors or attributes, they mainly include the physical condition of the soil, like whether the seed is damaged or not, whether the seed is entire, because we know that if a seed is damaged, then it may expose its endosperm to the atmosphere, and majority of the nutrient or the food material that is required by the seed to germinate is present in the endosperm. So if endosperm is exposed, then obviously it will cause degradation of the food material or the nutrients that are stored in endosperm, and that will eventually affect the germination rate of that particular seed, thereby affecting the yield of the crop and the quality of the crop. So first important factor is that the seed should be entire, there should be no physical damage to the seed. Second, the seed should not be suffering from any kind of notable disease, because it has been observed that sometimes if seeds are not stored properly, they may develop certain bacterial or fungal infections. And if you are utilizing such kind of seeds, then obviously the crop that will be obtained will be of poor quality, or it will be more vulnerable towards these kinds of infection. So one has to be careful regarding selecting the quality of the seed, specifically in case of physical factors. Now, the physiological factors are mainly attributed towards its germination rate, the viability of the seed, and the seed vigor. Higher the germination rate, more will be the yield of the crop. So if you are having a good quality seed with higher germination rate, obviously you are going to get a good yield of the crop. Then the viability of the seed. Now, it is largely defined by the ability of the seed to withstand various kind of infections during the growth, various kinds of changes in the environmental conditions when they are growing. So if a seed is having good viability, then obviously the crop will be more resistant towards any kind of the change in the environmental conditions or any kind of pest infections or the attack. And seed vigor is nothing but the sum of all the properties that are required for the proper germination of the seed. So seed should also have a good vigor. The third one is the genetic attributes, and these are mainly concerned with the purity, genetic purity of the seeds. So if you want to have the same variety of the crop, the seed should be genetically pure. Further, the genetic attributes also define whether the seeds are resistant toward various kinds of diseases, various kinds of attacks that are made by pest. So if the seeds are genetically strong, then obviously they are going to produce more strong crops, more robust crop, and those crops which have good secondary metabolite production. The last factor that determine the quality of the seed is storability. And the better seeds or the good quality seeds have good storability because they are least affected by the change in the temperature conditions where they are stored. They are least affected by the change in humidity conditions where they are stored, because humidity is a condition that will probably increase the chances. If high humidity is there, it will increase the chances of bacterial or fungal infection on the seed. Vis-a-vis, increased in temperature of the storage will also impact the germination rate of the seed. So if you are using a good quality seed, then the seed will be resistant towards such kind of humidity or temperature variation, and the crop that you will obtain from such kind of seeds, again it will be more resistant towards different infections. So this is how you can select a good quality seed by studying these attributes. Now here are a few examples where the quality of seed has where we can see the quality of the seed has its impact on the yield of the crop. I have taken an example of digitalis, and it has been observed that a robust crop with higher yield is obtained when heavier seeds of digitalis are used. Now this is more or less a physical attribute of the seeds. So if you are using heavier seeds, then you get a robust crop of digitalis. On the contrary, if you are using a lighter seed, then obviously the crop will not that robust and you will get a low yield. So one has to be very careful when you are using seeds as a mode of propagation. Now there are certain seeds which require specific treatments, or they have a slow germination rate and they require certain treatment to overcome that slow germination rate. And various treatments are, say for example, we sometimes soak the seeds overnight in water. Normally we grow coriander in our kitchen gardens, and we have observed this practice that we normally soak the coriander seeds in water for overnight, and this practice is being followed in order to soften the testa of the seed, that is the coating of the seeds, which enhances the germination rate and it fasten up the germination rate of those seeds. There are certain seeds which are more resistant or have slow germination rate, and they can be treated with certain growth hormones like 0.2% solution of gibberellic acid for 48 hours. There are certain seeds which require more drastic conditions or more drastic methods to increase the germination rates, like henbane seeds, that is seeds of Thymus niger, and they are required to be treated with sulfuric acid in order to increase their germination rate. Similarly, certain seeds require scarification, that is the crushing of the seeds or rubbing of the seeds on the rough surfaces in order to remove the testa from the seeds. And jalap seeds are normally scarified before they are being sowed, and jalap is nothing but Ipomia purga. So this is how you can also manipulate or also increase the germination rate of the seeds, and depending upon the type of the seed you are using, you can expose that seed to various treatments in order to increase its germination rate. Then, as we discussed, storability is a factor that that help in determining the quality of the seeds, and because the storage of the seeds also affects the germination rate, as we discussed. And here are a few examples that cinnamon and nutmeg seeds, if they are stored for more than 1 year, they significantly lose their germination rate. The germination decreases considerably. Likewise, colchicum corms, if they are sown fresh, then they have exhibited significant large or significant high germination rate. But if they are stored even for a shorter duration, their germination rate considerably decreases. On the contrary, there are certain seeds which are more robust and they can withstand longer storage duration. Say for example, Dhatura ferox and digitalis seeds, they remain viable for even on storing for years. So depending upon the type of the seed you are using, depending upon the type of plant you want to propagate from the seeds and its characters, you have to be very careful regarding the storage of those seeds. Now you have prepared the soil according to the plant requirement, you selected the suitable seeds for the specific plant. Next is the sowing of those seeds. It has been observed that if the sowing time is not appropriate, that adversely affects the quality of the crop that is being obtained. In a study, it has been found that yield of the essential oil obtained from chamomilla plant increases substantially if the seeds are sown in spring season rather than if they are sown in the autumn season. So one has to be again very careful regarding the sowing time also. Next we have is the method of propagation, and we know that plants are broadly propagated by two methods. One is the sexual method where seeds are used, and other is the asexual method where different plant parts are used for its propagation or for its growth. Now depending in sexual method where we use seeds, and depending upon the characters of the seeds, again you have to select the sowing method of the seeds, that is the propagation methods of the seeds. And depending upon the type of the seed, we have different methods like broadcasting, dibbling, drilling or line sowing, and transplanting. Now broadcasting is a method that is largely used for small sized and lightweight seeds, and those seeds which cannot germinate if they are sown in deep soil. And in broadcasting, we randomly scatter or we randomly spread the seeds on the soil. And the seeds that are sown by broadcasting method include linseed, isabgol and sesame seeds. So these are small size seeds, these are lightweight seeds, and if you sow these seeds deep in the soil, then the germination rate drastically decreases and it will affect overall yield of the crop. Now dibbling is a method which is largely used for the seeds which are having average size or mid-sized seeds, and they have a average weight or mid-sized average weight, I will say. And in dibbling process, we normally dug small holes, and in those holes 4 or 5 seeds are sown and they are covered with mud or the soil, and at a suitable distance again second hole is dug up and 4 or 5 seeds are again sown in that hole and it is covered with soil, and this practice is followed throughout the field. And the seeds that are sown by dibbling method include castor, fennel and papaya. Next we have is drilling or line sowing. Now in this, again this type of method is largely used for mid-sized and mid-weight seeds, and in this a line or a row is dug with the help of suitable equipment and seeds are sown in that particular line at suitable distances. And wheat and bajra are the crops that are normally grown by using this type of broadcasting method. And the fourth one is the transplanting method. In this method, seeds are initially grown under the nursery conditions, under the controlled conditions of say temperature, say light or water conditions, and once those seeds are germinated and the seedlings attain a specific size or specific age, then those seedlings are then transplanted into the fields. Now this kind of methods help in initial development of the plant when it is initial development of those plants, I will say, which are more vulnerable during their initial growth stages. So transplanting method is used for such plants wherein the seedlings are first protected under the nursery conditions and then they are transplanted into the fields. So friends, this was all about the factors related to soil, related to the seeds, the sowing time, and now the method of propagation, in which we have discussed different propagation methods under sexual propagation. So in the next lecture I will start with the asexual methods, and we will discuss that how different asexual methods are being utilized in order to cultivate or in order to propagate the medicinal plants. Thank you.